Every organ in the human body consists of tissues from these four categories: epithelial, connective, nervous, and muscle. Epithelial and connective will be emphasized at this point in the semester. For now, when we get to them, we'll just touch on nervous and muscle. Both nervous and muscle have their own content areas later in the semester. So hopefully you remember from the beginning of the class that a tissue is a group of cells performing common function. So epithelial first. Function-wise, epithelial tissue is boundaries, coverings, and protection, as well as secretion and absorption. Epithelial tissue is like the outer surface of your skin. It's the tissue you find on the surfaces of organs and lining tubes. So it's the inside lining of your heart, the inside lining of all your blood vessels, the inside lining of your reproductive, urinary, and respiratory tracts. It's also the surface of your skin, and it would be the surface of those organs. So inherently, because of its position, it's a boundary and a cover. Specific kinds of epithelial tissue contain many layers of cells, so they're protective. And then those cells that line a tract tend to do secretion and absorption. We'll look at pictures of them later. Characteristically, you have to understand what makes epithelial. Epithelial tissue is avascular. Your book's going to say something slightly different. Avascular means no blood supply. Now, warning, epithelial tissue is highly regenerative. It does a lot of mitosis and replaces itself pretty easily. So that means it has to be in contact with the blood supply, and it definitely is. The blood supply is immediately underneath the tissue. So you won't find blood vessels inside epithelial tissue. You will, however, find blood vessels immediately deep to epithelial tissue. Also with epithelial tissue, you are dealing with shapes of cells in arrangements. There are 3 shapes, and then there are 4 arrangements, and we'll look at those in a moment. Lastly, epithelial tissue has orientation. If I draw a line that represents a structure called the basement membrane, that's the deepest surface of epithelial tissue. Then we would have cells, maybe one layer, maybe more than one layer, but we'd have cells. Let's put some nuclei in our cells. Okay, and then what we can talk about now is the surfaces. This surface touching the deep basement membrane is the basal surface. This surface in contact with the environment is the apical surface. Now in the basal surface, again, those cells are going to be sitting on the basement membrane. And just so you know, the basement membrane in and of itself has layers to it. Now, the basement membrane under light microscope is very thin. So most of the times you won't see it. You'll see where it is, but you can't actually see the membrane itself or its layers, but it does have sublayers. And those layers are basal lamina and reticular lamina. One of the layers is structural and supportive, and the other is much more like a filter. Lastly, on the apical layer, if cells in this tissue are going to have cilia or microvilli, those will be present on the apical surface. Makes sense because that's what's exposed to the environment. So let's go look at them. In epithelial tissue, you have, again, 3 shapes, 4 arrangements. If a cell in profile is flat, we call it squamous. If a cell in profile's length equals its width, we call that cuboidal. If a cell is longer than it is wide, so it's kind of tall, that's columnar. So those are the 3 shapes. Each of those shapes will be found in one of the following arrangements. If I have one layer, that is simple. If if I have 2 or more layers, don't even need to count them, just 2 or more, that's stratified. If I have a tissue and the cells of this tissue are variable heights, however, they're all in contact with the basement membrane, that is called pseudostratified. And the reason it looks like it's many layers is because the cells are of different heights and the nuclei fall on different levels. Then we have a special case of transitional. In transitional, it's a kind of stratified, but what happens is as you move through the layers, the cells change their shape, and when you get to the surface layer, the cells are very big and domed shape, very round on the top. Well, the beauty of that is this tissue has the ability to stretch. So let's go through them rapid fire. Simple squamous epithelial. 1 layer of flat cells. Here they are. You're looking at them in profile from the side. Simple squamous epithelia is as small and as thin as you can get in the human body and still have a tissue. So it is perfect for diffusion, filtration, and if you overlap these flat cells nice and tightly, then it's a great lining because it's a low friction surface. We'll find it in the glomerulus, a structure in the kidney where it does filtration. You'll find it in the heart where it's a lining. You'll find it in blood vessels where again it's a lining. And guess what? Serous membranes made of simple squamous epithelia. The structures you see here are structures called the alveoli, for plural, alveolus is singular, and this organ is the lung. Here, it's diffusion. Simple cuboidal. At first glance, you may go, well, I don't see the tissue at all. Remember, epithelial tissue isn't always on the surface. It can be lining a tube. So what you look for are structures that look like tubes. You know you're in a tube if the very center of this tube is clear. That tells you that there's nothing there. That's space or liquid. So we're focused on these cells, this 1 layer of cells here, in contact with the basement membrane. Can't see it, but you can tell that there's something in there. And they're definitely not flat, but they're also definitely not tall. So those are simple cuboidals. We find them in tubes in the kidney called renal tubules. You find these in ducts of secretory glands. We're going to find them in secretory glands in the ducts and the surfaces of the ovary. Simple cuboidal epithelia perform secretion and absorption.